Hi, this is Miss Litton, and this is my wonderful Period 3 AP Bio class. Say hi. Hi. They're so excited. And this is the end of Chapter 14 on biotechnology and genomics. And we have just finished up 2 sections, one on DNA cloning and biotechnology products. And so these questions that we're going over are referring back to that previous lecture. And then we're going to move on and discuss gene therapy and genomics. So here is your first question. It is a map. You guys just hanging on till tomorrow? Hmm? Some people have it due today, I think. Yeah. What are words you always want to look out for in questions? Always, only, never. Those kind of ones are, you know, be very cautious on those. I have that same shaker cup. My shaker cups, like some of them don't fit exactly. You know, like if I don't get the right one when it comes out of the dishwasher, like, that's when it spills.
I just use it because I like instant green tea powder, so if I need to go.
Oh, instant green tea powder. Okay, snowy egret, lesser goldfinch. We're taking too long here. Oh, not very much gong. 19% of gong. Look at your BioBuddies clicker right now. They'll probably tell you if they got it right. Give them a nice big gong. Otherwise, shame bell. Shame. All right. All right, so let's go through this. Do plasmids serve as vectors? Yes. So you should have definitely got A. Plasmids can carry recombinant DNA, but viruses cannot. Who are your 2 vectors? Plasmids and viruses. So both of them can carry recombinant DNA. Do they only carry a foreign gene into the host? No, they could carry the host genes into the host, right? Doesn't have to be foreign. And only gene therapy uses vectors? No, you will use vectors, plasmids, when you do your lab, okay? And we're not doing gene therapy, okay? All right, check with your BioBuddies, see what one they struggled with right now, because most of you did. Look, discuss, make sure they understand it now.
No. Didn't happen.
Did you win, Michael?
What? I won the last one.
Were you Anna's hummingbird?
Yeah.
Sweet.
Alan's
Anna's.
Supposed to be anonymous.
I'm Anna's, he's Alan's.
Odd.
Oh my.
Oh, which of these not eat it?
Oh.
No.
Why don't we need sperm?
I'm third.
Okay.
That happened.
Okay, that happened. His sense of self-worth, it's gone. Okay. Now let's focus on the content of the question. Why don't you need sperm? If you're cloning, it's what kind of reproduction? Asexual. Asexual, you don't need sperm. You're going to take a nucleus. Okay, you're not going to fertilize something. You're saying, I want this exact organism. If I want that exact organism, I need to take a nucleus or all the DNA of that organism and make more with it. There's no combination. There's no sexual reproduction. I'm not going to take half its DNA and half of another organism's DNA and put it together. Okay, check with your BioBuddy. If they got the shame bell, make them explain it to you right now so they understand why A is the answer. Both of you need to explain it, and both of you are shameful. All right. Now we're going to move on to gene therapy in general. Okay. You could just say there is ex vivo and in vivo. How are you going to tell the difference? Look at the picture. What? Well, you have to have a naked man, obviously, in both situations. But was it in vivo? Do you see they're just injecting it directly into him? But in ex vivo, they're removing some cells, mixing them with the virus, and then returning them. So the therapy took place outside of the life form, or the therapy took place within the individual. See the difference? Okay. Now, now I need the oldest bio buddy to be the ex vivo expert. Go ahead, oldest bio buddy, be the ex vivo expert. Go. All right.
You have to take it out and find stuff like basically put it back in. And it's altered cells and the genetics coded proteins from all the
All right, and now I need the youngest bio buddy to be the in vivo expert. Go ahead.
Instead of like modifier inside, and they will find this Go find what you
All right, let's get some notes down on gene therapy. Accepted therapy for the treatment of a disorder that has been used to cure inborn errors of metabolism or other disorders. Use genetically modified viruses as vectors. Viruses as vectors. In ex vivo, inserting genes into cells that have been removed from the body and then returned. Blue, give them some examples of some ex vivo gene therapy treatments. Go ahead, Blue.
So that would be like bacterial one.
Okay, in vivo gene therapy, the gene is directly delivered into the body. Directly delivered into the body. And Slate, please talk about their examples, the trials they've already done. Go ahead. Okay, now let's get our big picture here. If you have a disease, ultimately something's going to be wrong with your what? Your DNA. And so what you're trying to do is fix the DNA. Then you will have the correct proteins that DNA is coding for, yes? Now, here's an issue you want to think about. We have long time thought of the DNA as the holy grail. Like if we understood the sequence of bases in the DNA and we could fix that, then all would be good. And that's true, understanding that DNA, but you know one section of DNA can code for several different what? Proteins, right? And then those proteins, in order to make them different, what makes them different? If it's the same DNA, what's going on? What's being altered so that you have different proteins from the same DNA? Introns removed, right? And so who's doing the messing with the introns? Which ones are expressed and which ones aren't? Okay, now restriction enzymes are man-made, or restriction enzymes originally came from bacteria who were trying to restrict the growth of invading viruses. That's where they came from, okay? But you have lots of, remember we talked about all the different control sequences? Okay, is the DNA unwound? Is it methylated? Is it unmethylated? Is the histone group methylated or not methylated? Do you have a transcription factor? Who's activating the transcription factor? Are there any small interfering pieces of RNA that are binding onto that mRNA that are affecting its ability to be translated? There's so much more. So genomics is important, but there's a whole field of proteomics that is getting studied. What are you hearing that? Proteins, right? Studying the proteins, because there's more proteins out there than there is DNA. Right? That's an even bigger field. And trying to understand how this, that's huge. And in medicine, specialized medicine is going to be a huge deal for those of you who are thinking about going into medicine. Or let's say you don't like people, so you don't want to have to deal with a person, but you like the idea of medicine, maybe you're going to go into research on that. Now, how many of you have ever gotten a cavity in your life? Anybody gotten a cavity? Okay. So what do they shoot in your mouth before you get that cavity worked on? Numbing, right? Does anybody numb different? Anybody have to get extra shots of Novocaine? I do. And I didn't get my first cavity till I was 19. I was devastated. I couldn't believe I got a cavity. I was so pissed. But anyway, I got that cavity, and the first time they gave me that Novocaine, they're like, okay, yeah, you won't feel this. And when they went to go work on me, I said, you know I can feel that, right? Okay, we'll give you a little more. You know I can still feel that, right? You know, they keep coming back 10 minutes later, give me a little more. My sister's the same way. What it might do for one individual is different than how it works on another individual. People metabolize that numbing agent on my neuron differently. So if that can alter, think about the medicines that we have. Think about just the idea of dosaging. What do they base dosaging on right now? Your what usually? Age. And sometimes your weight. Okay? I mean, yes, that does help. You don't give a baby 5 Tylenol. Okay? Great idea. Don't do that. Okay? So you have that whole issue, but think about it. If you can't even numb my mouth, how do you know that that's enough of whatever you're giving me? Right there, have people ever woken up in the middle of a surgery? Terrifying. Okay? I can't even imagine. Okay? Terrifying. Okay? So, because you could have your sensory neurons still firing, but your motor neurons not firing. So you could feel it, but you cannot respond to let them know that you can feel it. Okay? Yeah, how about that? Now, so just something like dosaging is different. What is effective? The body chemistry. Medicine will become highly specialized. Maybe in your lifetime, definitely in your children's lifetime. When we talk about gene therapy now, it's like, oh, I might get this nose spray, maybe I'll get the virus. How efficient is it? How effective is it? But maybe when you're like an old grandma on a porch or an old grandpa or something on a porch, and somebody is sick, you're going to be like, oh, back in the day, we just had surgery and antibiotics. Damn it. You know, and so you can imagine, I mean, people, what they will be, we don't even know what they're going to come up with. And they're going to be like, oh my gosh, can you imagine? You know, it's like my eyes were wrecked, you know, when I was a child, so they just fixed my eyes. Done, you know. I was going to be a diabetic, but they just changed my DNA. I'm fine now. Okay? But not only would we be able to do it for treatments, think about the knowledge that is held out there. And if people could read your DNA to more accurately give you your treatments, could they also read your DNA to know more about you? Right? If you go to a job interview, when I came to interview here, whatever, 26 years ago, okay, do you think I told them every bad thing I ever did? Or how I have a mouth on me and I always just say whatever I'm thinking? No, okay? Right? But if there was something in the DNA, if it said that I was going to get Huntington's disease, let's say when I got into my 40s, which clearly now that I'm 50, I'm not getting, I don't have the gene for it. But if it's in my DNA, Do you see how if they could just, oh, would you like a cup of coffee during this interview? Oh, nice to meet you, blah, blah, blah. Then they take the coffee cup, take a sample of my DNA, a little swab, go run it through and say, oh, she's going to have Huntington's disease. She's going to cost our insurance here an arm and a leg. Let's not hire her. Okay? That could be DNA profiling, right? Profiling me based on my DNA. Who gets to know that information? That is something you are going to vote on in your lifetime. Who gets to know that information? Maybe you're dating someone and you make out or something like, and you get all kinds of their DNA. You know, you've been kissing them, and then you go to a DNA profiling station and they take a swab off your lips and they run their DNA, and I will show you through a microarray, through a DNA chip, where they take the 30,000 genes we have and literally have them on a microscope slide, and they light up if you have that type of gene. So I can go there and go, oh my gosh, she has like 15 family diseases. Oh no, I'm not available, sorry, busy. Oh, I'm out of town this weekend. Okay, break it off slowly. Okay, so who gets to know that information? Because we are on the cusp of being able to just punch that DNA in and going, results. Okay, so who gets to know? Not only for treatment, but that kind of information. So that kind of study is part of what's called genomics. Okay, genomics. The study of genomes, the sequence of base pairs, our genes, and the genes of others. Now, there was something when I was just starting teaching in 1989. You weren't even born yet, right? When were you all born?
2000.
Okay. Anyway, that's so funny. Okay. In 1989, just starting out, they were talking about the Human Genome Project. And they were talking that they wanted to have, they finally had the computer ability to go through and map every, they can't map every person on the planets, but, maybe from 100 people, take 100 samples of their DNA and look to see what the base sequence is. And you know what? 99% of our DNA is pretty much exactly the same. There are little differences and little differences in repeat units, and we're going to talk about that, but that was like a huge big deal, the Human Genome Project. Now, from that, and you can see there's several websites. Again, for my researchers, this may be something career-wise you want to go into. DNA sequencing, the Human Genome Projects, and look right here, implication of what? Genomics on medical science. How is that going to affect treatments? Okay. And then, okay, here are the different organisms they have sequenced in what year. So look to see what was sequenced around your lifetime. When were you born? What's been sequenced? The Norway rat, perhaps? Okay. Yeah, so you can see the different years and what was sequenced before you were ever even born, too. Okay, so then you make comparisons amongst the genomes, and this is actually a little bit later in your notes, but I'll remind you of this. So that's a chimp and that's a human. So you can compare the base sequences of a human, you can compare the base sequences of another organism and see how similar and different they are. In fact, we're going to be doing what's called the BLAST lab, and in the BLAST lab, there is a huge, ginormous data bank of different organisms and the sequence of bases in their DNA. And you could take a newly discovered species where you're not 100% sure where it's related to or where it fits in phylogeny, and you can take the sample of DNA from them, put it into the data bank, and have it go boom, and it will tell you how much of the DNA is similar, just like that. Okay? You will do this. I will give you 3 genes that are real genes from an organism that was discovered, and you will put it in. This lab, we've only been doing an AP. This will be like the 3rd year I think we've been doing this lab, the BLAST lab. It's changed every year. You know why? The data bank that it's housed in, they have more and more what? Information. So it changes the way you do the lab. Now, this is not something I can put in Google Classroom, the genes, because they're so huge, the information. It took a while, but I have a link to them finally on our SchoolWires webpage, and you'll go and get that and basically copy and paste it. It sounds, but you'll put a link to it in there so that you can input all that DNA information from those. And it's one organism, and you're going to take 3 different samples of their DNA and put it in there and see how similar and dissimilar they are and who you think they're most related to. Okay? So that's done now. So genome comparisons, now there's huge data banks to do that. But you can just look structurally. This is comparing a chimp's ear to a human ear. Now where you see a real big difference is here, the nose. Okay? All right, now the HapMap project, this is a project that's just looking at the differences among people, okay, of different cultures. And what we may develop, let's say, as a drug treatment, if you're going into pharmaceuticals, when they are doing their drug treatments, if you only try it on one type of people, right? Let's say you pick Chinese people, okay, and that's where you're doing your research. You're in China, okay? And you're doing all of your research there. Is it possible that they could have a different arrangement of DNA that affects the way they metabolize their drugs? Yes. Okay? So studying different groups and looking at similarities and differences in their DNA. Are you more prone to one disease than another disease? Are you a carrier for this or that? But not just the DNA, but the cellular machinery you now know has a huge implications on the way that phenotype gets expressed. Remember, it used to just be, I know the DNA, so I know the trait. But now we know about things like epigenetic inheritance that influences whether that trait is ever expressed. And maybe if you do get a disease, people get it different ways. Expressivity, we've talked about that, right? Because it's a combination of things. It could be this gene, and this gene, and this gene, and this gene. That combination of proteins that you get gives you really bad breast cancer. Whereas this way, it's more treatable breast cancer. Do you see what I'm saying? So that's where it will get specialized, because they'll be able to take, they didn't used to have the computers, that's called bioinformatics. And now, bioinformatics, where you apply computer technology to information like this, for studying DNA or studying all the different proteins, this is now a major you can graduate with in college. It's just bioinformatics and applying that, okay? And there are companies and corporations coming up in jobs that were never imagined before because you didn't have the ability to know that, okay? So studying differences amongst people as well. So when you could have a single nucleotide polymorphism where it's just a difference in one nucleotide. Now, remember how we called point mutations? So it may not be a mutation, it's just a difference. Okay? A mutation implies something went wrong and caused that difference, which it could have been ancestrally. Okay? But you could be perfectly fine functioning adult. But as a result of this, you could, the way you respond to some sort of drug treatment could be different as a result of that. Or it could be a huge difference, right? What could be like tremendously bad about changing just one nucleotide? Give me some examples of what could be really bad.
Bad proteins and function.
You know, why a bad protein? Tell me why.
Because proteins are specific. They need like specific amino acids.
Okay, that's getting closer. Yes.
The mRNA is transcribed incorrectly, which means that the tRNA will have the wrong anticodon, which means that the protein is going to be...
The protein could be wrong. Now, remember we talked about you could just have the wrong, you could have the wrong amino acid in that place and it could cause no problem, or you could have the wrong amino acid in there and it could be a what? Stop codon, or you could have the wrong amino acid in there and it folds incorrectly. So sometimes they don't cause a problem, but sometimes they do. Okay, let me make sure we're caught up on our notes. Go to genomics. Defined as the study of our complete genetic makeup. And Blue, could you tell them number 2, please? Go Blue, speak it. Okay, when it says that includes introns and intergenic sequences. Introns are things that we what? And intergenic is also in between the genes. If it's inside the gene, it's intragenic. Like if this is a gene that codes for something, inside would be intragenic. Intergenic is between the genes. Do you remember in 9th grade or foundations class when you studied the STIRs and the single tandem repeats? Okay, and then you were looking. Remember, it was kind of annoying probably at first, but you had 3' 5' end, and you had to go through and count up like how many CCTGs, right? And you had 2, they said alleles, and that's because you had a homologous pair. You have the mom chromosome and the dad chromosome. So you're looking, how many repeats do I have in the mom? How many repeats do I have in the dad? And you counted the repeats, okay? And that would be intergenic, in between sequences. So if you had 16 and 18, let's say, and then what you did to solve your murder, right, is you got the different pieces of evidence on there, and then you were matching how many had the same number of repeats. And if it was an exact match, you're like, oh, he was smoking that cigarette outside of her apartment. Okay, what's in the trunk? Okay, they were looking at not just one section of repeating units, but when they look, they might look at 13 different areas in your genome. And in those 13 different areas, if they have the same number, and in fact it's an exact match, then you know that is you, right? Then you can make that direct connection. So that's one of the things that you would study in there when it talks about introns and intergenic sequences. Sequencing the genome. The Human Genome Project took how many years? 13 years. And now 2 million base pairs of DNA can be sequenced in 24 hours. It changed. In the process of doing the Human Genome Project, the technology changed. Polymorphisms are small regions of DNA that vary between individuals. And SNPs are single what? It's right there in your notes. Single nucleotide polymorphisms. Many have no effect, others can cause disease. Now, functional genomics. So think about this. So genomics is, you know your human genome, right? You know your DNA, the bases. That is known. Functional genomics says, okay, but which of these are coding for what? What is actual active DNA that we use? Well, to know what DNA is active, you need to know what the, not the exons, right? So remember cap in the tail and cut out the introns before it leaves the nucleus. Take this mRNA and work it backwards. Do reverse transcription. We've talked about this, right? Do reverse transcription and figure out what the DNA is, right? So to know what DNA is active, look to see what mRNA you have out there. Work your way backwards to identify the DNA that is functional. So that's the next part. Now functional genomics, working backwards from RNA to identify genes that are expressed. Working backwards from RNA to identify genes that are expressed. Okay, and dark-shirted BioBuddy, have you identified yourself? Listen as the light-shirted BioBuddy explains to you why SNPs are so important. Go ahead, light-shirted BioBuddy.
Oh, it marks the gene. It's a single point. I don't know. Oh, they're basically like
all right, so if it's intergenic, like what you're seeing right here, it's outside of the gene, and are you going to be that worried about it? No, it's a spacer, okay, maybe it's regulatory in some way, but if it's intragenic, it's Oh, look, this one actually looks like the way they drew it, that's outside. But you understand what I'm saying, right? Okay, so if it's in the gene, then it can cause a big difference. All right. Oh, I gotta keep going. Okay, so this gives you an example, intergenic region right in here, and then if it was somewhere in here, that would be, tell me what's the name of that? Just like intragenic, okay, within. All right, now, light shirted just went. Dark shirted one, could you please explain this diagram? Go ahead.
I'll tell you
Is there something up there that is shocking to you? Yes. Exons are what percent? 1%. That's not very much. They say there's a 1% difference between us and a chimp. And if we only express 1% of our DNA, that would be huge. Oh, yes, I will loan you. Who will take a class picture? Go. He will take a good one.
Go, Michael.
He's repping the class right now.
Quick. You just lost. Let's just tell him you lost on 5. All right. No, we're not like that to our students. We really do love you, Michael. Michael, I'm making a heart right now. You can't see it. Am I making a heart? Yeah. Oh, good. All right. Now, we have, and just so you know, so you don't get confused on here, we talk about exons as being expressed, and everything else we would refer to not as an exon, but a intron. Of those introns, I don't know if you can read that. It says introns are 24%. Other intergenic DNA, what does inter means? Between the genes, right? That bothers me. What are transposomes? Jumping genes. What? 45% can jump around and there's like literally no problem. What if some of them jump in the middle of my 1%? Okay, that would make me nervous, but apparently we're working. Large duplications. Those large duplications could be failed attempts at evolution, where you have copies of whole regions and you have more than one copy of it. It could be the basis of some of our evolutionary changes that we have, sources. It would probably be better, instead of starting from scratch to build a new gene, is to use a gene. We just asked 5 clicker questions. Sorry. You came in last place. You missed them all. Sorry. All right, now. It would be easier to take a functional gene and just change it a little bit rather than developing a whole new gene from scratch. Are you with me on that? People who make computers, do you think they start from no computer at all? No, there's some baseline operating system, right, that they work, and they're just manipulating it in some way. Same thing here with genes. Those could be those large duplications. And then look at these, simple repeats, okay? Those could be those repeating, those stirs where you're like CCG, CCG, okay, are the rest of it. Now, on your notes, structure of the eukaryotic genome. In general, more complex organisms have more complex genes with more and larger introns. Intragenic sequences are within a gene. 95% plus of the average human gene is introns, just everything that's not an exon, okay? Introns are now considered as regulators of gene expression. Regulators of gene expression. So intergenic sequences are DNA sequences that occur between genes. Types. Okay, so these are all the different types. We just saw a graphic on that. Repetitive DNA elements are common. 2 or more nucleotides repeated many times. And it constitutes nearly half of the human genome. A tandem repeat, sequences are next to each other on a chromosome. Think STIRs, short tandem repeats. Interspersed repeat placed intermittently along or among chromosomes may play a role in evolution of new genes. evolution of new genes. For example, a repetitive DNA element known as the ALU sequence, do you see I have that in there as an example, is interspersed every 5,000 base pairs in human DNA and comprises nearly 5 to 6% of total human DNA. Same sequence, every 5,000 base pairs. Okay? Because of their common occurrence, interspersed repeats are thought to play a role in how new genes could come about. Transposomes. What do we know transposomes as? Jumping genes. Ability to move within and between chromosomes, sometimes acts as a regulator, could have an evolutionary role. And then unique non-coding DNA may actually create small-sized RNAs. Remember how we talked about small interfering RNAs? Remember that? Okay. Small-sized RNAs that are more effective than proteins at times. Okay, so now that we have talked about that, okay, when we say, when we were after what makes us who we are, all we were looking at was the how, what percent? 1%. Because we were saying, these are our 30,000 genes approximately we have that makes us us. We were looking at that 1%. What about the other 99? It could be regulatory. It could be sources of evolution. It could play jobs and roles we don't even know yet. And then this makes us then revisit the central dogma. So you and your BioBuddy, if I say central dogma, what are the 3 parts to the central dogma? Discuss with BioBuddy. What's the first one? Starts with an R. Replication. And what is that? DNA to DNA is replication. Okay, what's the next one? Transcription. And that is saying now I'm going to use this DNA to code for what? RNA. Yeah? And then what's the third part? Translation. And the translation assumes that I'm using the RNA to code for proteins, and those proteins make me what? Me. But maybe that's not the end game all the time. I've got 99% of the DNA. I got 99 problems and ain't one. Okay? Maybe that's not the end game. Is because only 1% of my DNA is even coding for proteins. Maybe the end game is forming RNA that then controls the other 1%. Maybe that's part of my end game. Maybe that's what I'm just trying to get is some RNA, some messenger RNA that's not going to be translated into a protein, but it's going to be a signaling unit for something else. Okay, and that's what scientists are looking at and trying to open up. It's not just the DNA that we're after anymore. So revisiting the definition of a gene, expansion of central dogma. Expansion of central dogma. Maybe put more emphasis or emphasis on the RNA generated. On the RNA generated than the actual DNA. A gene product may not be a protein, right? Our gene product may be some kind of RNA. Gene product may not be a protein. Some prokaryotes have RNA genes. Think about it bigger. RNA is what? Single-stranded or double-stranded? Single-stranded, okay? But when you have something that is single-stranded, you run into the problem that something could what to it? Bind. Maybe the only reason we have DNA is just to store the important RNA. Think about that for a minute. Maybe the only reason we have DNA is to store the important RNA. And that you make a DNA copy that then binds, right, double-stranded, and it's protected. Do you see that? And I can store it where? In my warehouse, the nucleus. But really the action is the RNA. And when you look at evolutionary history, you look back at what came first, DNA or RNA, you know where it looks like it all points to? RNA. You know why? RNA can act as a storage for genetic information, and it can act like an enzyme. Because what came first? right? The DNA or the protein it makes, because you need the proteins to synthesize the DNA, right? And DNA then codes for proteins, the chicken and egg. What came first, right? You can't make more DNA unless you have DNA polymerase binds to that promoter. What is DNA polymerase? I know, it's a protein. So how do you make more DNA if you gotta have a protein to do it? RNA. Maybe RNA came first. Okay? You seen expanding? All right? So it's, and for so long, all we cared about was the DNA. Now, science is looking at all the different aspects of it. Okay? So, now, functional and comparative genomics. Functional genomics says its aim is to understand the exact role of the genome in cells or organisms. The exact role of our genome in cells or organisms. Well, one way to do that is to do these profiling, this genetic profile. Okay? Now, I'm going to show you a real one, but I just want to explain it to you first. Okay? This right here, we have like 30,000 genes. What if I take all that DNA and I've got 99% that I've got to ignore, right? So I've got to ignore that DNA, find the 1%. Now I want to amplify that 1%. What is a procedure I could do to amplify that 1%? PCR. And I want to amplify certain genes. I don't want the intergenic, right? I don't want the in-between genes. I want the actual DNA that codes for a protein. So maybe I work backwards using mRNA to find the DNA, yes? Then once I find that, I have to put primers on either side of it, mark that region off, so that I can do PCR, the polymerase chain reaction, in a thermocycler, make bunches of copies of it, right? Then I'm going to take it and I'll make it single-stranded. I could use some heat or something, right? And make it single-stranded. Cut up those functional genes, and I could put single-stranded DNA on a computer chip or a slide. Okay? Basically, I have all the genes that actually code for proteins, if we're going central dogma style. Okay? And then I could take somebody else's, I could take Kate's DNA, and I could find those same regions, amplify it, cut it up, and take her DNA and flood it on this marker. But let me tell you what this marker is. It only contains mutated genes, diseased genes. If her DNA, any of her DNA, is complementary to the DNA that's on there, what does that mean? She's going to get that disease probably. Do you see that? She's going to get that disease because it binds to it. Okay, and it lights up. So let's see that right here. Let's do it where I am a pointer. I'm gonna show you that one. Okay, what one is the name of that one? What's on a microarray? Remember that one. I want to show you how to make a microarray. Un momento, por favor. Oh, here it is.
We built a very simple robot that used very simple and ancient technology, the technology of the fountain pen. And what the robot does is it takes a set of fountain pens and dips them in each of these little ink wells in this 96 well plate. And the ink in these ink wells is the DNA that represents a human gene. Each well represents a different human gene. The pens dip into the wells, and then they move over and print the DNA ink in a nice array on a little microscope slide like this. And they repeat that process over and over again from hundreds of these plates, finally producing an array that represents about 30,000 different human genes. And then we can use these microarrays in a very simple experimental procedure that allows us, that basically causes the spots to light up whenever the genome in a particular cell or tissue sample uses one of these genes, one of these words in the script that it's writing.
All right? He has human genome on a flipping microscope slide that he printed with DNA ink. Okay? Now he could take normal human gene, do that, or abnormal, right? And if you had abnormal DNA on there and it lit up, that means your DNA has that, and just like that I can find out about you. Again, I could find that out and use it for good and how the best way to give you some medicinal treatment, or I could use it for, yeah, I could find out about you, maybe information about you, okay? So this is underneath DNA microarrays, DNA chips, current cellular mRNA binds to DNA on a chip and can know which DNA is active. A genetic profile can indicate genetic illnesses and what drug therapy might work best. And what drug therapy might work best. And then comparative genomics, we already looked at that, like when we were comparing us to the chimp, and I told you could do that with a new species and plug it into that database to find out. Comparative genomics, your aim is to compare the human genome to the genome of other organisms. It's used for testing, studying changes, and evolutionary relationships. Okay, proteomics, you're studying proteins. And again, now a whole industry just about studying proteins. Businesses that are looking at that. Let's see if I can do, Resource Center. Proteomics. Okay, so now they have all of these ways to use and analyze and identify the different proteins that are functional within a cell. So this could be used for like medicinal research, etc. Let's write some notes about that one. Defined the study of the structure, function, and interaction of cellular proteins. A proteome is a collection of a species' proteins, which is larger than the genome. Larger than the genome. And you can study the three-dimensional shape of a protein. Some of your parents might work at Amgen. It is the coolest thing. You go in a room, and when they're trying to analyze a protein, they have huge TV screens, and you wear the 3D glasses, and you get to move the protein around and look at all the aspects of it, because the way that works as a drug, as a medicine, you know enzymes have specific what? Yeah, okay, good. What do we call that area where it binds to the substrate? Active site. So the drugs and the way they work and impact your body, their three-dimensional shape is huge. And if you're trying to mimic some cellular component to cell that let's say something you're missing, they have to get the shape of it exactly right, otherwise it won't bind. So they just look and study the three-dimensional shape, and they'll turn it this way and that way, and you're studying it wearing those glasses. It is super, super cool. Okay, so that's one thing you would do. Goals, there are two of many. Oldest bio buddy, tell them the two of the goals that are right there for proteomics. Okay, and that I told you, you could now, this was never, when I was in school, this was not a major, okay, that you could do. But now there's one, and this is the University of Michigan, and this is the Department of Computational Medicine and Bioinformatics. Okay, so they have a whole huge department just dedicated to that. And bioinformatics is applying computer technology and what you can do now with databases to analyze things like proteins or to analyze the DNA. And here they have a whole graduate program, a medicinal program as well. So go to bioinformatics. Youngest bio buddy, is it your turn? Yes. Go. Do number 1. Okay, and then the blast lab, I told you we're going to do a blast lab, basic local alignment search tool. It is a computer program that can identify homologous genes that code for the same proteins among genomic sequences of model organisms. So it's a search tool, and you're comparing genes among model organisms. Discoveries that have been made, gene function, copies of genes can be correlated to disease. For instance, if you've heard of Fragile X syndrome, and this, who do you think Fragile X syndrome is going to hurt more, a male or a female? Male. Why? 1X. And if they get too many repeating units, the chromosome doesn't hold together and it literally starts to break off on the bottom. That is the Fragile X syndrome. So it's how many repeating units do they have. So it can be correlated with disease. And then oldest BioBuddy, tell them what metagenomics is. All right. So all of this, like I said, is underneath when you think about DNA technology and what we can do with that. All right, here we go. It's a map. Remember what kind of words you look out for? I left Michael because you missed the last 5, so I'm feeling bad for you. Let's go house wren, snowy egret. What are words we look out for? Only, none, never, right? Okay, just checking. Oh, 75% of you are getting a gong. 25%, bell of shame. Serve it up if you need to. If they don't show, they didn't know. All right, so it is still investigative and makes use of viruses to carry. You wouldn't want to pick B. Why don't I want to pick B?
Because it has some.
No success. Okay, well, it has some success. In fact, you actually looked at some of those examples. Is only used to cure genetic disorders. I don't like those words that say what? Only. Yeah. This is a map as well. I'm not saying it's a man, but it is a map. That looks like a really bad word. Oh, 50% of you get the gong. Show your clicker. Show your clicker. Good job, California.
Let's not look at yours.
All right. A lot of you got B. Okay. It is not able to enter cells. Okay, viruses have to be able to enter cells, right? You know that. That's how they work, by hijacking the cellular machinery. Yes? Yes, you can. Did I say anything about eliminating unnecessary steps? No.
So that's at the
Oh, to the right of the phone.
Oh,
And D, if it prevented infection by other viruses, don't you think we would all get it done? You would never get sick from a virus again. Okay? If all else fails, just use logic. Okay? Pretend like, okay, approach a question like, I know nothing. Let me just use my logical, my large mammalian brain, unless you left it at home, I understand. And just try to answer it that way. Okay? Just it's a strategy.
Michael, relax. I forgot everything I learned. I have no clue. I'm just gonna guess. I'm gonna fail with my fingers. I'm gonna just show him.
Let's go, peeps. Will we get 100? What are the two vectors? Plasmids and what? Oh, okay, good. Plasmids and viruses are your two vectors. Protoplasts. Some of you are going, protoplast plants. Yeah, I've heard of that. A protoplast is just plant cells without a cell wall. Okay? Those are more easily available than two vectors to go into them. They're not the vector. They can accept a vector. So here's a cautionary tale. The more you know, the more those distractors can be distracting. Right? Because you're like, oh, I've heard of protoplasts. Yeah, it has something to do with plants. Okay, yeah, that's right. Okay? You need to think through. Always go back and go, what are they asking? What are they asking? And then go back and look at that. Because you know viruses and plasmids are your two vectors. Okay, check with your bio buddy because only 23% of you are getting a gong right now.
20%.
75%? Shame.
I'm at 20% right now.
Okay, the end. All right, get out your next set of group shared notes. Chapter 15, go, go, go. You're super smart.